Name That Microbe by Miranda, Skylar, Karen, and Rachel. In today's presentation, we will be going over bacteria, virus, protozoa, fungi, and lastly will be our references.
For our first topic, bacteria, I picked Fusobacterium nucleatum. Fusobacterium nucleatum is an oral bacteria and is a normal eukaryotic microbe to have. It can, however, grow out of control and cause diseases. Fusobacterium nucleatum, abbreviated to F. nucleatum, is an oral bacteria, but it can cause problems in other parts of your body. F. nucleatum has been associated with preterm birth and has been known to cause other complications during pregnancy, such as stillbirth and hypertension. Fusobacterium nucleatum is an anaerobic oral bacteria that is known to cause many different diseases in humans, such as periodontal diseases. It is gram-negative, which means it does not retain the crystal violet stain because of its wall structure. Microscopy tip. To actually see bacteria moving around, you will need a microscope with at least a 400 power magnification lens. If you have even higher lenses than that, you will be able to see a lot of great detail in the bacteria, but it can be hard to focus on them that close up because they move around.
Herpes simplex virus is a very contagious virus that is widespread throughout the U.S. Herpes simplex virus is divided into two types, HSV-1 and HSV-2. HSV-1 is typically transmitted by oral contact between individuals and is associated with oral herpes. HSV-2 is usually transmitted sexually and is typically associated with genital herpes. The HSV-1 and 2 infections manifest sores or blisters, usually on around the lips or genital area. This virus is highly contagious. Some studies have suggested that up to 65% of the U.S. population is infected with one form or the other. Herpes simplex virus 2 is the second most prevalent STD known to date, with an estimated 24.1 million infections. There is no cure for HSV infections. However, there are antiviral drugs that can help control outbreaks. The herpes virus consists of a core surrounded by a capsid. The capsid is icosahedron in shape. The capsid is composed of 162 capsomeres. The capsid has an outer envelope with multiple proteins. Microscopy tip. The envelope surrounding the capsid is removable by chemical treatment, which will allow for better observation of the genome.
Protozoa is the next category of microorganisms. They are eukaryotic, single-celled organisms that exist as parasites or as free living. They come in various shapes and sizes and can be found all over the world. Giardia lamblia is an example of a common protozoa. Giardia lamblia exists in two forms, cysts and trophozoites. Cysts are the inactive form of the protozoa. They are intermittently and for indefinite periods of time excreted in the feces of both symptomatic patients and asymptomatic carriers. Trophozoites are the active vegetative form that replicate in the small intestine and cause the uncomfortable symptoms of diarrhea and malabsorption in humans and many types of animals. Both forms are unicellular eukaryotes. However, the trophozoite form is also flagellated. These flagella are the appendages that allow the protozoa to swim and move throughout the intestines. These parasites were first discovered by Van Leeuwenhoek in 1681 while he was examining his own diarrhea samples. Later, they were identified as being pathogenic and a cause for diarrheal disease. Giardia lamblia spreads through food, water, surfaces, and objects, but the most common method of transmission is by swallowing contaminated drinking water or water from pools, lakes, or rivers. Forms of the protozoa have different microscopy. Trophozoites are pear-shaped and have either one or two transverse, claw-shaped medium bodies, as seen in the picture on the right. They both have visible nuclei, well-developed cytoskeletons, and endomembrane systems. Flagella can also be seen. However, they do lack mitochondria and peroxisomes, which are common amongst most eukaryotes. The inactive cyst form of the protozoa is an ovoid to ellipsoid shape. The immature microbes have only 2 nuclei, while the mature ones typically have 4. Microscopy tip. The best technique for observing cysts under a microscope is by using iodine staining. To best observe trophozoites, using trichrome staining is best according to the CDC.
Up next is fungi, Penicillium notatum to be specific. Penicillium notatum introduction. It is commonly known as Penicillium chrysogenium. Penicillium notatum is a species of fungus in the genus Penicillium. It is commonly found in mild temperatures or in subtropic regions. It can be found on salted foods, but is mostly found in indoor environments such as damp or water-damaged buildings. Penicillium notatum is non-pathogenic to some individuals, but it can cause mycosis in individuals who are immunocompromised. Mycosis is when the skin is invaded by the fungus. Treatments for this infection include multiple antifungal agents. Some ways to prevent the growth of Penicillium notatum is to keep the area dry and well ventilated. If you happen to notice signs of the growth, it is best to soak it thoroughly with a non-toxic registered fungicide. The morphology of Penicillium notatum is filamentous hyphae with conidiae, and they appear to be blue-green with yellowish pigments. Penicillium notatum history. Penicillium notatum is an important fungi because it has evolved into an antibiotic, which is now known as penicillin. In 1928, Alexander Fleming was the first to discover penicillin. He was experimenting with the influenza virus at the time. Fleming returned from a 2-week vacation to find that a mold had developed on an accidental contaminant, Staphylococcus, culture plate. Upon examination of the mold, he noticed that the culture prevented the growth of Staphylococci. This is now known as penicillin. Since then, the discovery of penicillin has changed the course of medicine and has enabled physicians to treat formerly severe and life-threatening illnesses. Microscopy tip. The best technique for observing fungi under a microscope is by using a slide culture.